What's up, everyone? Welcome back to the channel. Today, we are going to be talking about one of the most important biomolecules in all of biochemistry. Today, we're going to be talking about carbohydrates. Now, by definition, carbohydrates are polyhydroxyaldehydes or ketones or substances that yield them upon hydrolysis. That basically means that a substance that has multiple OH groups, polyhydroxy, and also contains an aldehyde group or a ketone group is classified as a carbohydrate or a substance that breaks down to yield any of these. Now, carbohydrates are primarily composed of three main elements: carbon, hydrogen, and oxygen, and they often follow a general molecular formula CH₂O. Carbohydrates may also contain other elements like nitrogen, phosphorus, and sulfur. But at basic level, we would not be talking about carbohydrates that contain these. Now, carbohydrates serve a number of functions. The most basic function is as a source of energy. We all know from basic science that carbohydrates are energy giving food. So this is because of their ability to provide energy for maintaining a number of bodily processes. Now, carbohydrates can also be a form of energy storage. As some forms of carbohydrates, such as glycogen, can be stored in the muscle. Carbohydrates also have structural role, such as their part to play in the plant cell. It's a carbohydrate known as cellulose. Now, carbohydrates have a number of other important functions, but these are going to relate to the basic biochemistry that we'll be discussing as we go on. Next, we're going to be looking at how we can classify carbohydrates. Now, as mentioned before, carbohydrates may have one of two functional groups, the aldehydes or ketones, but we'll get back to that. Now, we will be talking about how carbohydrates may be grouped according to their size. Now, according to size, carbohydrates may be monosaccharides. They may be oligosaccharides, and they may be polysaccharides. Now, a monosaccharide is a sugar that contains just one sugar unit. An oligosaccharide has 2 to 10 sugar units, and a polysaccharide has greater than 10 sugar units. Now, by the way, the word saccharide is Greek for sugar. So now we'll be looking at some examples. The most common monosaccharide is glucose, composed of 6 carbon, and it is a single sugar unit, so it's got just one sugar. As you can see, oligosaccharides have 2 to 10 sugar units. And an example is sucrose, which is made up of glucose and fructose. Okay, so fructose is another 6 carbon unit, but it has a different cyclic shape. We're going to get into this as we go on. Now, because it has just 2 sugar units, this oligosaccharide is known as a disaccharide. We're also going to get into this in more detail. And a disaccharide is made up of two sugar units joined together by something known as a glycosidic bond. So this is the glycosidic bond joining glucose and fructose together to make sucrose. The polysaccharide is made up of greater than 10 sugar units. Okay. And these sugar units are also joined by glycosidic bond. So they may be made up of monosaccharide units that are similar, like in the case of starch, you have repeating glucose units where glucose binds to glucose, binding to glucose, and so on. They may also be made up of different monosaccharides binding to themselves. We'll look at this in more detail as we go on. So next, let's look at monosaccharides. The monosaccharides are colorless crystalline solids that dissolve in water, and they may be aldoses. They may also be ketoses. Now, at the very beginning of this lecture, we talked about how carbohydrates may be aldehydes or ketones or substances that yield them on hydrolysis. Now, an aldose, as the name implied, would contain an aldehyde group, and a ketose should contain a keto group. So the O's is just talking about the fact that it's a sugar. Okay. Now, the position of the carbonyl group, which is this, is what determines whether or not a sugar would be an aldose or a ketose. Let's look at this in a little more detail. So we have here the most basic aldose sugar known as glyceraldehyde and the most basic ketose sugar known as dihydroxyacetone. Okay. Now, if you look very closely, you would see that this is your CH₂O group, and this is your C double bond O, which obviously gives the sugar the name. Now, when the carbonyl group, that's this bit, is found anywhere at the very beginning or at the end of the structure, that is when it gives you the aldehyde group. That means that anywhere you see CH₄O or C double bond O at the beginning or the end, it is automatically an aldehyde group and therefore an aldose sugar. Whereas when it's a ketose, you find C double bond O anywhere besides the beginning and the end of the structure, anywhere in the middle. That sugar becomes a keto sugar or a ketose in this case. Now, if you look closely again, you'd notice that this aldose sugar is made up of 1, 2, 3. So therefore, it is a triose because it has 3 carbon atoms. And we already know that it contains the aldehyde group. That makes it an aldo triose. Now, this sugar, this keto sugar, also has 1, 2, 3. That makes it a triose, but in this case, it becomes a keto triose. So we have some other examples of aldoses that could be triose sugar, pentose sugar, hexose sugar. So when it's got three, that's one, two, three carbon, it's a triose, an aldo triose. If it had four, it would be a tetrose sugar, an aldo tetrose. Remember the aldehyde group at the end of each of the structures. So you can see glucose is a hexose because it's got six, one, two, three, four, five, six carbon, and it has the aldehyde group, making it an aldo hexose sugar. On the flip side, we also have ketoses following the same structure. When it's got this in the middle, dihydroxyacetone, we've talked about that. Ribulose is a ketopentose because it's got one, two, three, four, five. With the C double bond O anywhere besides the ends of the structure. Similar with fructose, which we talked about earlier, but this is the linear structure of sucrose. So just like that, you would have a ketotriose, ketotetrose, ketopentose, ketohexose, ketoheptose, ketooctose, and ketononose, and so on. So the number of sugar units determine the classification of that sugar. The number of carbon rather classify the name you'd call the sugar. Now let's go to cyclic structures and anomers, because if you notice in the structures that you just saw, all of those carbohydrates were in linear form. Now isomeric forms of monosaccharides that differ only about their configuration are known as anomers. And don't worry, I'm going to explain this in a little more detail. And any sugar with 5 or more carbon atoms tend to form cyclic structures in solution. That means that when the carbon atoms are greater than 5, they would form cyclic structures in solution. The two different kinds of cyclic structures they may form, they may look like this, which is a 6-membered ring. This is known as pyran. It may look like this. This is known as furan ring. So because we're talking about sugars now, a sugar that forms the 6-membered ring would be a pyranose, and a sugar that forms a 5-carbon ring would also be called a furanose. Now let's go back to how anomers work. When a sugar like glucose is dissolved in water, it may form two different structures. Okay, wherein the carbon 1 holding the H and OH are arranged like this, and you can see the OH pointing downward. When it forms this kind of cyclic structure, remember this is a pyranose. You can see the name pyranose. When it forms a structure like this, we call it alpha configuration. And when it forms a structure wherein the OH is pointing upward, we call that beta configuration. Okay, so the two anomers of glucose are alpha and beta glucose. Now, in a solution, eventually both alpha and beta glucose will interconvert in a process called mutarotation. Now, let's take a look at disaccharides. Now, as I've said before, disaccharides are a group of oligosaccharides made up of just two sugar units. Some examples include maltose, sucrose, which we saw earlier, and lactose, which is found predominantly in milk. Sucrose is found in sugar, particularly sugar cane. And maltose is found in, well, malt barley. Now, as I have explained before, any sugar with 5 carbon atoms and above tend to form cyclic structures that may look like this. Now, if you have two cyclic structures that look like this, a bond will form between them. This bond is called a glycosidic bond, and it is what holds disaccharides together. Now, this is exactly how a glycosidic bond is formed. As you can see in this example, this is alpha D glucose. Alpha because the OH is pointing down. And this is beta D glucose. Beta because the OH on carbon 1 specifically is pointing up. So this is beta. And this is alpha because of this OH on carbon 1. Now what happens is this OH on the first carbon and the OH on carbon number 4, because if you count from here, you've got 1, 2, 3, 4, come together, and the OH binds with this hydrogen, and when that happens, water is released. So what's left is just this O. So that oxygen atom, this lone oxygen atom, is what joins both structures together. Okay, so in this particular example, we have two glucose units, so they're identical. The only thing that differentiates them is the orientation of the OH group on carbon 1 that makes this beta and makes this alpha. So joining two glucose units produces a disaccharide known as maltose. But maltose is just the short name. The long name is alpha D glucopyranosyl 1,4-D glucopyranose. And we're going to talk about how we arrived at this very long, complicated looking name. So next we are going to talk about naming a disaccharide. Now, naming a disaccharide involves 4 main steps. The very first step is giving the configuration of the very first monosaccharide unit on the left of the structure. So if you've got 2 units like this, the orientation of OH on this very first one, let me use another color for that, is named first. Okay, so if the OH is pointing down, the very beginning of the name of your disaccharide would be alpha, and if it is pointing up, the beginning of the name would be beta. Okay, so you start from there. Now, the next step involves giving the name of the sugar as well as the nature of the ring. So, if the sugar, we're still talking about the very first sugar, that's this sugar, is glucose, for instance, a six-membered ring like this is known as pyrano. You are going to have glucopyranosyl. Okay, let's assume this first sugar, instead of it to be glucose, it was galactose. Galactose is another sugar entirely. We would call that galactopyranosyl. Assuming this very first sugar was mannose, it would be mannopyranosyl. Okay, so the name and the nature of the ring is what determines what follows next after the orientation. Assuming it's a five-membered ring, such as fructose, your disaccharide would start being named after the orientation, would be fructofuranosyl. Fructo, the name of the sugar, furan, the name of the ring. Okay, so that is the next step. Now, the third step involves where the bond forms between the two carbons, between the two sugar units, sorry. So remember, this is 1, 2, 3, 4 in this case, and this is 1, 2, 3, 4, 5, then the sixth one is up here. Okay. So if the bond is forming between here and here, this would be 1 with an arrow 4. Okay, because the bond is forming between carbon 1 of the very first sugar unit and carbon 4 of the next sugar unit. In some cases, you may have this structure flip the other way so that 1 is facing the other structure, and you now have 1, 1. So you'd have 1, 1. This is the next step in the name. That's why the name is so long. You started off by giving the orientation, and the next step, the name of the carbohydrate, then the next step, the nature of the ring, then the next step, the bond in between the two of them. Thereafter, you name the second structure. Okay. So if this second structure now, is another glucose, for instance, it will be gluco six-membered ring pyranose. But you would also take into account the orientation of the OH group on this side. So whether it's beta or it's alpha. So assuming it's beta, it will be beta glucopyranose. So let's look at this name together. You have alpha because the OH on carbon 1 is down, it's pointing down. You can see that's that's why the bond is able to form here. D glucopyranose. Gluco because this ring is glucose. Pyranose because it is a six-membered ring. 1,4 because as you can see, this is 1, this is 4. So 1,4. Okay. Then D, this particular example did not have beta, but there should be beta D, sorry, beta D glucopyranose. Okay, because it's glucose again with its six-membered ring, making it glucopyranose. And that's the final step in how you name your disaccharide. Disaccharides generally have short forms in how you write them. So glucose, short form for glucose is GLC in brackets 1,4 because that's where the bond takes place. Glucose again. So maltose is made up of 2 glucose units. So looking at some notable examples of disaccharides, we have lactose. And as you can see, lactose is made up of galactose, that's the galacto, and glucose. Okay, so beta because the OH is here pointing up. Okay, when it's pointing up, it's beta. Okay, so beta D because it's oriented to the right, but we didn't talk about that. Beta D galactose, this is the structure of galactose. Pyranosyl because it's a six-membered ring. 1,4 because that's where the bond is taking place between 1 and 4. Beta again because if you look at this structure, the OH of the second sugar is still facing upward here. Gluco because it is the structure for glucose, pyranose. Now the only difference between the structures of glucose and galactose is the orientation of the OH in carbon 4. In carbon 4, the OH of glucose is facing down, while that of galactose is up, in case you are wondering the difference because they look so much alike. So because of that difference, these structures are epimers, but we'll be talking about that in another video. So the short form would look like this: galactose beta 1 to 4 glucose. Another notable example is sucrose, which we've looked at before, but you can see that this is a furan ring. So it's a furanoside, and you can see that the bond is between 1 and 2, which gives it this 1, 2. Okay, so it's alpha because OH is down, D glucopyranosyl, 1, 2 beta because the OH is up. This is the OH in this case. Beta D fructo because this is fructose, that's the name of this sugar. Then 5 carbon ring uranoside in this case. Okay, so next we are going to be looking at polysaccharides. Now, polysaccharides are polymers of sugar units. That basically means that you have multiple sugar units bonding to themselves, like so. Some of these chains may branch off like this. In some cases, these polysaccharides may be composed of the same units. For example, if this is glucose and this is glucose and this is glucose and this is glucose and this is glucose. In some other cases, it may be glucose, fructose, glucose, maybe galactose, and so on and so forth. Now, when the polysaccharide is just a linear structure that does not branch off, and it is made up of repeating units of the same sugar, you can see that each of these is the same exact sugar. It is known as an unbranched homopolysaccharide. Homo means same. Polysaccharide mean poly, many, saccharide, sugar. Unbranched, same, many sugars. Okay. So repeating units of the same sugar that are unbranched is an unbranched homopolysaccharide. And if they branch off, then and it's still made up of the same units, then it is a branched homopolysaccharide. You may also have a heteropolysaccharide because they are different sugar units. So this may be fructose, this may be glucose, this may be galactose, this may be mannose, this may be tetralose, and so on and so forth. And it is in a linear form, so it is an unbranched heteropolysaccharide. And when it is branched, it is now called a branched heteropolysaccharide. A major example of a branched polysaccharide, starch, as well as glycogen. Now, the only difference between starch and glycogen are the differences in how much they are branching. Starch is much more highly branched than glycogen, and both starch and glycogen serve as storage forms of carbohydrate. Starch in plants and glycogen in animals. Heteropolysaccharides tend to form extracellular matrix in animals and bacterial cells, offering protection and structural support. So you'd find some of these in form of peptidoglycans in bacteria and animal tissues. In conclusion, carbohydrates are more than just sweet things we eat. They are essential to life structure, energy, and communication. And I hope this video has helped to introduce this topic. As always, be sure to give this video a like and subscribe. Thanks for watching. I'll see you next time.